Thanks, Dan. I hope you can see the slides.
Yes, thanks.
Yeah, I would like to thank the organizers for the opportunity to talk here. So I'm going to focus on atomic model validation for cryo-EM data. So just to give you a brief background, so most of you are aware that the cryo-EM area is progressing towards better resolutions, thanks to the resolution revolution in the last 8-9 years or so. So we're getting more and more high resolution structures from cryo-EM, and as you can imagine, more people are building models, and you have several thousands of atomic models deposited in PDB, which are derived from cryo-EM data. So as you can see in the plot here, it shows the trend in resolution with each year. So you can see that the best resolution structures, best resolution is getting improved until 2020. And also the average resolution of the cryo-EM structures is also improving. Still, if you look carefully in the plot, you can see that the average resolution of the cryo-EM structures are still slightly worse than 5 angstroms, between 5 to 6 angstroms. So this is again the resolution distribution of cryo-EM maps in EMDB. So if you look at the distribution, you see that the number of structures better than 5 angstroms is roughly half. And if you look closely into the segment, you can see that majority of this segment of the distribution is belong to 3 to 5 angstrom, and there's only a bit less than 1,000 structures which are better than 3 angstrom resolution. So I mentioned this, that the number of fitted models are increasing year by year, and currently there are around 6,500 fitted models derived from cryo-EM data. As you can see on the right, these fitted models are associated with data from different resolutions. So majority of them are from better than 4 angstrom maps, which is expected, and there's quite a large number between 4 and 8 angstroms as well. So atomic models are built in a wide range of resolution data here in cryo-EM. So it's really important to give care to the validation and the details in the structure built. So as you can imagine, the features interpretable at different resolutions are different. So here is a table showing what features we can ideally interpret at different resolutions. So if you're looking at low resolutions, we can identify the broad shapes, for example, the domains or the subunits. If you go to finer, better resolutions, you can interpret better and finer details of the structure. So secondary structures better than 9-10 angstrom resolution. And when you enter better than 3 angstrom resolution range, you can look at side chain conformations, and even best resolution structure so far is around 1.5 angstroms. So you can really look at the details of atomic positions. So as in the case of crystallography in cryo-EM as well, we can look at different aspects of validation, so fit to data, model quality, overfitting, and model bias, and so on. So I'm going to talk more about the first two areas here, so fit to the data. There are several metrics used in the cryo-EM area for estimating fitness to the map. And the most widely used metric is the cross-correlation coefficient. And as you expect, so if you get higher values, it means better fit to data. But the value by itself, the absolute value of cross-correlation by itself can be a bit ambiguous, in the sense that the example that I show here, it's the same model with the map, and the map is processed differently in different panels of the figure here. And you can get different cross-correlation values just as a result of processing the map differently. And if you use a low resolution map, with the same model, you get significantly different number there. So the cross correlation is kind of quite sensitive to how we process the data and what is included in the calculation. And the other metric would be equivalent in the reciprocal space. So the model map Fourier shell correlation. So it's another widely used metric in the cryo-EM field. So we look at the Fourier shell correlation in different resolution shells. And an average of this can be calculated, which is called FSC average, which is a weighted average of the Fourier shell correlations in different resolution bins. And so FSC average is a single number, so it's a global indicator of fitness of the model to map. And there are a few metrics which can estimate local quality of fit of the model. One of them is the SMOC score, which is equivalent of cross correlation, looking at per residue fitness to the data. And here's an example from the SARS-CoV-2 structure. So it's an RNA polymerase structure. Here, on the top right corner, you see that the side chain fit and the density here is not perfect or not great. This is a result of a sequence register error in the fit. So the sequence model here is out of register. And if we remodel this, for example, this is done in with Buccaneer ab initio model building. And if you remodel this, you get the right sequence and the right side chains in the right place. You see a better fit of side chains in agreement with the map. And this can be identified with this local estimates, local fit to map. You see a dip here corresponding to the sequence register error. And the other aspect of validation, which I'll talk about, is model quality. So most of you are aware of mol probability. So mol probability is, again, the most popular model quality estimate in the cryo-EM area as well. And it's quite useful. So here are some examples which highlights that if you find errors in the same area of the model, these errors can be related. For example, here you find clashes, and if you flip the side chain, you can avoid these clashes you find here. Again, the cis conformation here introduced a clash with a side chain in the neighboring amino acid. And again, if you model it in a trans conformation, you kind of rectify the clash here. So the outliers may be related. So if you're trying to fix one outlier, you might end up fixing more around. Again, looking at the cis and trans conformations in the model. So this is roughly the kind of expected numbers in PDB. So the cis proline are more prominent than cis non-prolines. And if you're modeling a non-proline, so make sure that you have the map information to justify the non-proline, the cis conformation that you model. So here's an example of a cis peptide here modeled, and the density justifies the model conformation. And this is another example where the model conformation is cis, but the density doesn't really justify that. So there are a number of cases. If you look at cryo-EM models, you will find a number of cases of cis non-prolines model. So here, the other examples, you can see that there's no map information to explain this modeled cis conformation or the model outlier. So this have to be taken care of. Again, Oleg gave a nice talk, the previous talk about Ramachandran Z-score. So just looking at the number of Ramachandran outliers is not quite useful in some cases. So here is an example where you can be in the favored or the allowed Ramachandran space, but still you can move around and distort the hydrogen bonding network in the secondary structure. Here, it's a beta sheet here. If you rotate the carbonyls, some of the residues here in the beta sheet, you break the hydrogen bond network, but you can still be in the allowed Ramachandran space, which you will not notice if you're just looking at the Ramachandran map outliers. So CABLAM is a method developed by Jane Richardson group and co-workers, so Chris Williams and Jane Richardson. And it essentially tries to address this problem. So it looks at the quality of the backbone, looking at pseudodihedrals. So these are, for example, consecutive C alphas, the dihedral between consecutive C alphas, or the dihedral involving the carbonyl atoms and so on. So this is a set of favored conformations again in both 3D and 2D space based on these pseudodihedrals. And if your model regions fall in the favored space, like how we look at the Ramchandran maps, we can identify outliers here and so on. So here's an example where you identify outliers. So the 3 carbonyls facing the same position, which is an outlier here in a beta sheet region. And CABLAM identifies such issues in the structure. So overall, just to give a summary for this part of the talk, so we need different quality metrics which look at both fit to data, model geometry, overfitting, model bias, and so on. So we essentially need a number of different metrics which are ideally quite complementary to each other, looking at different scale of features when you use models coming from different resolution data. And EMDB model challenge is very useful resource for looking at different metrics. So it's basically a challenge where different model building tools are compared. So different groups take part in the competition. They build models for a specific target, and the models are compared based on different metrics. And the metrics are also compared and so on. So it's a very useful resource. And the data there is also useful for other method development. So currently we developed an interface in the CCP-EM software suite for validation of atomic models from cryo-EM data. So here's a list of tools currently available. The general idea is to integrate more and more tools here, which are complementary, looking at different information. And it gives information on fit to data and the model quality. So here's model map FSC plots, and on the left there's tables on the mole probability and CABLAM outliers, the FSC average values, and also the local fitness to the map. So per residue plot of fit to the map scores. And also you get a table where the outliers are clustered in space. So if you find large clusters, it essentially means that there's probably a region with serious issues in the structure. And it's probably a better idea to focus there first. It's quite common in cryo-EM structures to have a large number of outliers, which is expected working in slightly low resolution areas. But this sort of clustering will point to serious issues in the model, which needs to be fixed first than looking at other individual outliers. And there's a link to COOT interface there where these outliers can be fixed. So you get a list, a window with a list of outliers where you can fix these outliers. So I'm just going to a slightly different analysis on validation of SARS-CoV-2 structure. So I
have maybe 1 minute or two, Agnel.
Okay, so just to highlight that, so this is the distribution of mole probability scores versus resolution. So you have a different range of resolution, and if you look at mole probability scores, this is a spread of different mole probability scores. So sometimes people tend to refine more on the geometry side rather than fit to data. So it's a general take-home message is that it's important to have a balance between the two. So here's a plot between fit to data metric versus the mole probability score. And here, if you have a better fit to data and better mole probability score, that's probably the area, the place where you should be. So there are a number of structures of the SARS-CoV-2 complexes where you have poor fit to data and better mole probability scores, for example. So here's one example where the model is mistraced here, at least one part. There are other cases as well, slightly low resolution map, where the model fit to data is not optimal. Again, just to stop with this resource. So this is a resource looking at validation of SARS-CoV-2 structures derived from cryo-EM data. So it's a web resource accessible from here. So here we try to map mutations in the SARS-CoV-2 viral genomes and map it onto the 3D structures from cryo-EM data. And currently there are more than 28,000 sequences, and the number of mutations that we have is 13,000 odd. So we try to give distribution of these mutations in different countries in the world and look at validation scores for the structures where these mutations can be mapped. This interactive interface where you can look at the mutation site and the structure, and also look at validation statistics. So here there are some geometry outliers in the mutation site and so on. So it basically gives some indication of the quality of the model at the mutation site. And based on this, you can interpret interactions and so on. So you also get a list of interactions. So I'll stop with this and acknowledge all the collaborators and funders. CCP4 has been very helpful in setting up the CoVal service, the COVID structure validation service. And I'll stop with that. And thanks for your attention. I take any questions.